artificial intelligence will completely transform our world. But what is AI? Why will it affect you? And how can you and your business survive and thrive through the AI revolution? Welcome to AI and You. Here is your host, author, speaker, and futurist, Peter Scott.
Hello and welcome to episode 322. I'm talking with Mohammed AlQuraishi, professor of Columbia University's Department of Systems Biology and a member of their program for mathematical genomics. DeepMind's AlphaFold led me to be interested in the problem and impact of protein structure prediction, and Mohammed is one of the top researchers in the world in computational modeling of that. The lab that bears his name is focused on biological perspectives at the molecular and system levels. He has an MS in statistics and a PhD in genetics from Stanford University and was a departmental fellow at Harvard Medical School. Last week, we talked about Mohammed's background in molecular biology, the big goals of that field, and his North Star of understanding biology well enough to simulate it. Why I haven't yet seen the kind of revolutionary impact I was expecting from AlphaFold, what it means for a protein to fold, and why it's a big deal, and how AlphaFold and similar models work at figuring that out. This week, we'll talk about how the advent of AlphaFold landed in the protein folding community, how AI is about to transform molecular biology and scientific experimentation, the possibility of simulating a cell, OpenFold, which is Mohammed's answer to the closed-source parts of AlphaFold, and his vision for the next 10 years of the field. So let's get back to the interview with Mohammed AlQuraishi. I think many people just on the outside of the field, reading the headlines, would not have realized that predicting proteins was something that had been going on for a while anyway, and AlphaFold was not the first entrant into that field. It was just the best one to date. You've blogged on this and written some very candid and engaging statements that lift the lid on the field and in particular, the emotions of the people involved there. Part of that was talking about people who were already working in the field of predicting protein structures for a while. And then Google, DeepMind, I'm not sure which one it was at the time, came along and disrupted it by just jumping to the head straight away. And you wrote, quote, "In a delicious twist of irony, we, the people who have bet their careers on trying to obsolete crystallographers, are now worried about getting obsoleted ourselves." And that's that fear that many people that would recognize in all kinds of fields that AI is coming for their job. I don't think that they did that, but your blog reads like you're reporting on some kind of competition. It's as though this heated battle between different factions that are like chess grand champions. And the paraphrase of what you wrote there, I wrote down, is Google beat out Big Pharma, which you also ranted about devoting thoroughly inadequate resources to the research in the first place. So now that seems to put the energy back into what Google did. So can you just settle for us, what was the size of that contribution relative to that field? I read some biologists quoted as saying, we don't believe these results, but we have no choice. They seem to check out. In terms of the impact, maybe you can liken it to something that gives us on the outside a sense of how big a bombshell this was.
Oh, it's certainly a big bombshell. So you're absolutely correct in what you said. The field certainly existed for many decades, and many people were in it, myself included, and remain in it, going after these harder problems now. I mean, I think one way I like to think of it is I think they compressed something like 10 years of progress in like 2 years or something. My sense is we would probably... It's a little bit hard. It's interesting. Looking back, so AlphaFold 1 came out in 2018, AlphaFold 2 in 2020. So I think maybe if science had proceeded at a trajectory it did back then, I think it's conceivable to say if AlphaFold 1 and 2 hadn't happened, then we would not have AlphaFold 2-like systems, till, 2026 or 2028. Now, or a few years from now, instead of back in 2020. Of course, the big intervening thing that's happened in the middle is AI. And the way at which it has progressed. So it's conceivable that the timelines would have shrunken anyway, just because of AI. But that is at least, at the trajectory of the time. That's what it felt like to me, and I think what it felt like to a lot of other people. Another way of saying this is, I would say the field was maybe, inching every year, improving, I don't know, a couple of percentage points, 3 points, 3 points, 4 points, something like that. And then in the kind of the AlphaFold 1 to 2 gap, we moved up like 30 points in 2 years. And there is that competition, actually, I should say. So there was this, and there still is, this event that happens every 2 years where people get together, put their best models forward, and try to predict structures in a blind way. So it's obviously, in academia, there's competition always because different groups are working on similar problems. But this was actually formalized in this kind of biennial competitions that happened every 2 years.
Oh, AI coming into that kind of like ImageNet, perhaps. What's that competition called? Is it still going?
A CASP, the critical assessment of protein structure.
And does that still happen?
It still happens. Now focus more on harder problems, like the corner cases we were talking about earlier, mutations, drug binding, so on.
And the analogy that came to mind for me, and perhaps it's overblown, was one of people being launched on an interstellar space mission that's going at 40% of the speed of light, and they're going to take 10 years to get to a star. And 2 years out from their destination, a faster than light ship that's been launched from Earth a week earlier pulls alongside and says, "Do you want to ride?" Which they could be grateful for it saving them 2 years. On the other hand, some kind of frustration and resentment would be understandable. Did AlphaFold land in the field something like that? And have they gotten over it?
Yeah, I think that's about right, actually. And I would say most people have gotten over it. This is more of a statement about kind of the culture and society of scientists. It's, on the one hand, of course, we're invested in the work we do. And so you always want to see your work or your colleagues' work, your friends' work succeed and be the thing that grabs the headlines and so on. So that's one aspect of it. But on the other aspect of it, of course, we're also part of this greater enterprise. A lot of us got into this space, into the exercise of science, because we think it's important for humankind at large. And so when you set aside the ego and then look back at the fact that the field has moved forward because of this development, of course, you can't help but rejoice in the fact that this has happened, right? So it's often this kind of slightly bittersweet thing where you feel like, on the one hand, I'm very happy that this has happened. This is a good thing for the world. But maybe for my own little corner of it, now I have to think hard about what do I do next, or that sort of thing. And then I think this was certainly, at the time, in 2020 when this happened, I think this was definitely a kind of feeling that pervaded many scientists working in this space. I think the nature of progress is such that I think it didn't take very long for people to figure out new things and new problems to work on. And I suspect, now as a society, we're sort of going through this whole exercise again, much more broadly, with AI emerging in a much more general sense, certainly in mathematics and computer science, and likely in other fields to come. So I think this is sort of thing which will only become more prevalent.
Well, you've mentioned a few times about how you're working on harder problems now, and AI is developing into platforms like agents where you can put together, chain a whole bunch of steps and have it work through them, which makes me think of the steps that a lab would undertake in carrying out an experiment. What role does AI play in molecular biology at the moment? What analogy do you draw? What kind of metaphor do you have for how it fits in?
So maybe just one point of distinction to make, which is important, but maybe slightly subtle, is certainly everything we've done up to this point, including things like AlphaFold and so on, are, you could say, exercises in AI instead of more narrow AI, often called machine learning, right? So in that regard, it's permeated essentially all of computational molecular biology, let's say. But for the most part of the flavor of human-designed models, right? So AlphaFold was very much, the architecture of AlphaFold was very much a product of human ingenuity, even though it's trained on data. And so the specific kind of model weights and the way it behaves is derived from, this kind of gradient descent exercise. I think what you're referring to now is a bit of a different thing, where AI is general and where it's beginning to inform the design of the experiment, the execution of the experiment, and so on and so forth. Maybe even at some point, the choice of experiment, the questions to go after. I would say in that sense, these are actually still comparatively early days for AI in molecular biology. Like programming, for instance, or mathematics, where already we're seeing, very serious problems and very serious workflows and tasks getting automated. That actually quite hasn't yet happened. There's not been an AI that has developed an AlphaFold. That hasn't happened yet. But I don't think it's very far away. Given the scale of things, the speed at which things are moving, I wouldn't be surprised within 6 to 12 months, we'll probably see models designed by AI that are actually tackling serious questions in molecular biology. So when I say early days, this is all in these very extremely fast timelines. Yeah, so that's where I would say we currently are at.
When you undertake experiments, do they look like computation performed on existing data, or do they look like going to a lab and getting your fingers dirty with some kind of biological material?
It's primarily the former, so taking existing data, but that data may not necessarily always correspond to data, say, either in the public domain or data that is just existing. So, work with partners to generate data effectively on demand with an explicit purpose to train machine learning models. So, we ourselves don't get our fingertips dirty or whatever, but we certainly engage with people who do. And I think certainly it feels to me like the field moving forward is going to increasingly have that flavor. In a way, the low-hanging fruit is developing models built on existing data sets. That's there, you could just do it. It's a purely computational exercise. But I think as AI gets better and better, is going to kind of pick all these long-hanging fruits. And so what's going to remain effectively is the stuff that requires much more intentional coordination between the data generation side and the modeling side. And I think that's where things are increasingly going to be heading.
I'm wondering about what the steps are in your typical workflow for conducting experiment. If it involves data and data products and data analysis, then if that's a lengthy process, then the bottlenecks are presumably human orchestration. And the AI agents naively offer the potential for automating that orchestration in a way that could radically accelerate it. Is that something that's possible or that's happening or not?
I think it depends a bit. I think this is happening in some cases, but certainly, particularly in biology, the problems are often speed limited by biology, right? Particularly if you think of things like cellular biology, where you're depending on something to grow, let's say. Less so for chemistry, where it's things you could sort of, put in a test tube. I would say in the current situation, we are still bottlenecked by the speed at which biology and chemistry happen. So essentially the kind of physical processes that are underpin these things. So that still remains a bottleneck, more so than orchestration. It is still, however, the case that experimentation is becoming increasingly high throughput. And then I actually think we are not far from a day where we could essentially design experiments or have agents design experiments that are then run essentially in cloud laboratories, executed, and then the results are retrieved. So you could see how all these various components, from the design of the experiment, the execution, the retrieval and analysis, become increasingly more automated and essentially, become an agentic workflow. So I think that is and will happen. My guess, in the limit of sort of, if you want to put it this way, sort of in the limit of infinite intelligence, or at least in the limit of intelligence being sort of saturated, so in the limit in which additional intelligence doesn't buy you anything, my guess is we will get bottlenecked again, are by physical processes, right? It's possible, not by any means guaranteed, but it's possible, for instance, that a mouse just takes several months to grow to get to the state that you wanted to get to. And maybe we come up with some clever ways to speed that up. But there may just be kind of inherent limitations, physical, biological, chemical, that would ultimately be the bottleneck. So that would be my guess, looking forward in terms of where progress may be slowed down.
And heading in that direction, you've said that an AI-powered cell simulation might be less than 15 years away. Can we start talking about that by you comparing the size of the problem of cell simulation to the size of the problem of protein folding?
Yeah. So it's a good way to think about this problem. So on the one hand, you could think of this, but I think this would be misleading, but you can think of this strictly as spatial scales, right? So maybe proteins, are on the order of a few nanometers to tens of nanometers in sort of size, while cells are the order of micrometers to hundreds of micrometers in size. So, that's sort of roughly the scale. Then maybe you say, oh, it's 3 to 6 orders of magnitude bigger. It's that, right? It's just purely kind of more volume and more things going on within that volume. So that's one way of thinking about the problem. But I think it's a bit misleading because I actually think it understates the problem. And the reason why I say that is because, proteins, comparatively speaking, again, when we go back to this coevolution business and so on, we have, in retrospect now, enough data to have sort of solve the problem or, solve a version of the problem. But there is really no analogous situation whatsoever for cells, right? Certainly for the resolution that I would like to be able to kind of build a cell simulator. So it's not merely a matter of spatial scale, it's a matter of technology, even in principle, being able to image and quantify the state of cells in anywhere near the resolution and fidelity that we can do today for proteins is a big sort of challenge, right? It's a huge gap that currently exists.
But what about the time domain? I understand protein shape doesn't change, but a cell is kinetic. It's got all kinds of movement going on inside, right? What does that do to the problem?
Right. So that's very analogous to kind of the spatial thing I mentioned, right? So proteins do change in shape depending on their function. Some of them are fairly rigid and very static, but some do change. But often these changes maybe span, let's say, the scales of, let's say, milliseconds, maybe in some cases seconds, but that time scale. And I should say even that scale is inaccessible to us. So we can't really simulate proteins, generally speaking, greater than the scale of microseconds. So, a million less than a second. While certainly the lifetime of a cell, could be minutes, and certainly organisms, could be days and years and so on. So there, again, you have several orders of magnitude gap between that. I think all of these are challenges. I don't want to overstate one over the other, the spatial, the temporal, and the kind of technological in terms of this fidelity question. But somehow, if you were to, and this isn't a particularly informed answer, but if you were to put a gun to my head and ask, pick one of these as being the biggest, I actually think it's the third one, the fidelity question, because we just don't even have the tools yet. The other stuff you could just imagine, if you have enough resources, maybe scaling things up across time, across space. But the technological problem feels a little bit more, let's say, scientific, more of an open question as to what even, what would it like to be able to, again, measure and quantify cells with the same fidelity as we can do for proteins? That feels almost like a scientifically open question to me.
And it feels like one of those life's work kind of goals there, with just the size of the problem, but also the potential for what solving it means. I want to go back to, you mentioned OpenFold, and that's something you created in 2021. Can you introduce us to that and why you did that?
Yes, sure. So when AlphaFold 2 came out, at the time, I should say DeepMind was incredibly generous in a way, having put all this investment in developing the system and essentially releasing what one would call the kind of inference code or the prediction code, which means you could use the code that they released to make new predictions for new proteins. What was missing, however, was the training code and the training data sets. So you could make predictions, but you cannot reproduce the model. You cannot make new models on top of it. You cannot extend it in various ways. It's a bit analogous, I would say, to the current kind of controversy over open weight models like Kimi this week. That is an example of a model that is open weight, so you can make inferences with it, you can make predictions with it, but you can't actually extend it in any way. So it's better than, say, the fully closed models from OpenAI or Anthropic or what have you, but it's not as good as having something that is really a foundation on which you build, on which you can build. So we thought we want to fix that, right? Because so what OpenFold was essentially a kind of a perfect reproduction of AlphaFold, but one that also includes the ability to train the model from scratch. It included the training data that you need to actually do that as well. And so it became more than just kind of a fixed artifact that you could use to make predictions, but it sets up a platform, something you could then use to build new capabilities and new foundations on. And that's been really a kind of at the time when we built it, it was just sort of a side project. But now it's become one of the biggest things that my lab focuses on, and it's sort of acquired this whole consortium around it of companies or what have you that support the effort.
And where does hardware come into this? When you run AlphaFold, is that running on Google's hardware? And what hardware does OpenFold run on?
So when you're making predictions, generally speaking, you could run it on Google Colab, which is effectively Google's cloud, but you could also run it locally on your computer or on your local cluster, what have you.
Is that AlphaFold you're talking about there, or OpenFold?
Both.
Okay.
Both have that property of being runnable wherever. Initially, actually, when AlphaFold 3 was first released, you could only run it on their servers behind a firewall, but eventually they sort of came around and decided to put out the prediction code. So you could run it anywhere. Now, the training procedure is a bit different in the sense that you could, in principle, run it anywhere, but it's much more expensive. It's much more computationally intensive. And so that's typically the kind of thing that you can only really do in high-performance computing environments.
And what is the cost of making a prediction in terms of hardware use, either AlphaFold or OpenFold?
It depends a little bit on the size of the system you're trying to predict. If it's a simple protein, individual protein, could be a minute on a GPU. So it's a 60th of a dollar, a couple of cents, fairly cheap. On the other hand, if you're predicting a fairly large complex, it could be a couple of hours on a GPU or even GPU cluster, which could mean, several dollars for a prediction.
But a far cry from what it costs to do with crystallography. I mean, just not in the same universe.
Absolutely.
When you mentioned that OpenFold could work on as few as 3,000 inputs in the training data, what's the motivation for doing that? Is it to have smaller models that cost even less?
Primary motivation was scientific. We wanted to understand essentially the question that you raised. How much data do we need to train a model of this sort? Like, how is this model working? How data hungry actually is it, right? So that was fundamentally the question we wanted to ask, essentially curiosity driven. But it has at least one main practical implication, which is to say, okay, we've sort of solved protein structure, or at least partly solved it. But what about other types of modalities? Let's say nucleic acids. RNA have three-dimensional structure, quite interesting, quite functionally relevant, very dynamic. The existing models don't do a very good job predicting that structure. So we think our experience with OpenFold and understanding the kind of the data hungriness of this problem points the way, provides something of a recipe or at least a kind of a blueprint in terms of what types of data and how much data needs to be collected for other similar problems to be able to achieve the same degree of fidelity that AlphaFold has done with proteins.
Who are the principal customers for protein structures? Who takes the output of these predictions and says, thank you very much, and uses that for something?
I would say primarily academic groups trying to understand the function and behavior of proteins of interest, and pharma trying to drug these proteins, trying to modulate their function. Those are probably the two big groups.
Does that result in faster vaccines, for instance?
It has the potential to speed up the early stage of drug discovery. I mean, you were just pointing to this earlier, right? That the crystal structure is very expensive, very labor intensive, and so on. So now imagine you have a set of candidate molecules that say, bind your protein of interest. So they're promising, they're lead targets. And you want to determine experimentally the structure of each one. That is labor intensive. That's going to take your structural biologists a fair bit of time and money to do. Now you could at least make predictions, see the predictions are predicted to be reliable, and then based on that, perhaps triage your set of experiments to a narrower set that is more targeted to the problem of interest. So at least I would say it lowers the cost and speeds the rate at which early stage drug discovery happens, not late stage, not animal trials, not these kinds of things, but that first set of tasks that companies engage in.
I'm thinking about an instance where AI was involved in constructing chemicals according to various goals, and a researcher decided what happens if we flip the bit that says we want this to have a positive outcome, and it generated a whole set of horrifying toxins. Is there a dark side potential to this work where there are things that could be done with it where you would say that needs to be locked behind a guarded door?
I think eventually, yes. I would say the bigger risk is more with design methods rather than prediction methods, if that makes sense. Right? So ones that actually produce new compounds or produce new proteins, those have the potential capability to start producing pathogenic compounds. I would say the current risk is still comparable to sort of conventional methods because there's often the many steps downstream of that initial generation that have to happen, and those are the bottlenecks. So I don't yet think it's become a risk. I would imagine that in some years from now, a few years from now, we would get to the point where the capability frontier shifted so much that this does become a concern. As you know, for instance, with Anthropic, there's been all this discussion about Fable. Fable is very crippled when it comes to biological questions. We didn't even answer basic questions about like the mitochondria or something. Very skittish.
Wow.
But I think we will have to navigate that at some point, right? Obviously that's silly, but things like, make me a protein that is highly toxic, maybe that's something that it should refuse. And you can't do that today, truthfully, with any degree of accuracy. But if that becomes a possibility a few years from now, obviously that does become a consideration.
When we talked about simulating cells, we crossed another line there in going from protein to cell, which is, to my mind, going from something that isn't alive to something that is. If we look at the bigger picture here of what an organism is and whether life could be ultimately understandable through computation, Do you see yourself on a road towards engaging with that question? What does that mean to you?
To understand life or cell as computation?
Or using computation to help understand life?
Yeah, absolutely. I mean, I think that is the North Star of my sort of scientific mission. So one way, maybe just to kind of put this a little bit in a more crystal form, what is it that I think is really interesting here is using computation to understand life. But I think that the dual of this is understanding life as computation. And I think computation can help with that. So to me, what cells ultimately are and what they do, they're effectively probabilistic programs. They're programs in the computer program sense of the word, that sense stimuli internally and externally and integrate that information to execute decisions, to do things, ultimately including to divide. So it's very much this sort of living program that's free living in the world and that's doing things. But fundamentally, the key underlying processes are all computational in nature. It's all computation, right? It's all information processing. So that to me is very attractive because it provides almost a different paradigm for how computation can occur. We've had computer programs for a very long time that are human engineered. We are now having computer programs that are learned from data, neural networks and what have you, LLMs and so on. And then life is sort of a third paradigm, another type of program. So that's very exciting because it sort of provides really a kind of fundamentally new into the question of computation. I started earlier saying physics is deep, biology maybe is not so deep. But in this particular instance, I think it is because it's sort of getting at the kind of one of the fundamental sort of processes that actually happen, this notion of computation. So that's very exciting. And I do think computation can help us do that because I like physics. Biology is very complex, it's very heterogeneous. You have a large number of particles, each of which has own personality, interacting with each other. And it feels like it's the kind of thing which fundamentally can only be amenable to computation to really make progress on. So I think that they sort of go well together, those two aspects of the problem.
I'm being prompted here to think about this in terms of architectural patterns, like that protein would have certain patterns in the way that it folds itself. There are certain configurations that make sense and many, many more that don't. And that a cell has its own set of architectural patterns and the way that it works and maintains its membrane integrity and things like that. And then life has some more familiar architectural patterns of like reproduction and excretion and digestion and things like that. So we're creating at each step these new tropes that describe the way that this thing works, its characteristics. As we wrap this up, what are your thoughts for how you would like to see the field developing in the next 10 years?
Good question. It's a hard question. I think there's maybe a tendency in science in general, and I mean, it's a human tendency to try to get the answers quick. And particularly for, answers having to do with human health, it makes sense. The urgency is there. People suffer from all sorts of diseases and, we want to cure them as much as possible, of course. But I raise this because what sometimes worries me in a way, maybe let's say the kind of the orientation of science in general, is to elevate the practical over the kind of, let's say, the fundamental. And why this is important is because the fundamental questions, the more basic questions that don't have immediate practical relevance, often in the long run prove to be quite important for lifting our scientific understanding and lifting humankind as a whole. So what I would like, to answer your question, the way I would like to see biology evolve in the kind of the era of AI is, on the one hand, of course, have a strong focus on translational applications and the ways in which it can improve human health and disease. But at the same time, also be kind of cognizant of the potential that can be had when biology is understood at a very fundamental level. And do that. And essentially apply AI, deploy AI, and our own ingenuity, going back to the beginning of this talk, and essentially trying to ground biology in as firm a foundation as physics has had the privilege of being grounded in for the preceding century. So that feels to me like what, for me, would be the ideal outcome for the next decade of biology.
Great note to end on. Love this conversation. Could clearly keep going for a long time, longer than it would certainly be polite to keep you here. It has been an absolute pleasure here to engage with your mind on these big questions, to experience just how far your imagination and passion takes you and answer my stupid questions in the process. So thank you very much, Mohammed AlQuraishi, for coming on AI and You.
Thank you for having me.
And that is the end of the interview. I think it's just amazing that I get to talk with someone who's working on how to simulate a whole cell. And you get to hear that too, which is something more people should get to hear. And, you've heard me say this before. The way for them to find out about it is for you to hit the like button on your podcast app, or better yet, write a nice interview. If there's someone you know who would be interested in or benefit from the kinds of conversations and interviews we have here, please tell them about the show. They're not going to find out about it otherwise. In today's news, ripped from the headlines about AI, Demis Hassabis, the founder of DeepMind, now Google DeepMind, wrote a manifesto calling on the United States government to establish a new AI watchdog with the power to screen the world's most advanced models and to coordinate an industry-wide slowdown if the dangers mount. The manifesto, titled A Framework for Frontier AI and the Dawning of a New Age, calls for a more systematic approach to AI regulation, funded by the industry and answerable to the US government. He wants this to be set up within months, before the end of 2026. Good luck with that. Specifically, he proposes a standards body modeled on the Financial Industry Regulatory Authority, FINRA, and says that Frontier Labs would share their models with them up to 30 days before release for safety testing. As you might expect in such a volatile world, the reactions to his proposal were mixed, with some applauding it and others accusing him of promoting regulatory capture, which is certainly at least a side effect for the proposal. But are we at a point where that's unavoidable as part of a responsible approach? That sets up next week's episode rather well. I'll be talking about the existential risk and other long-term thinking around AI, which continues to evolve in ways that are of interest and concern to all of us. That's next week on AI and You. Until then, remember, no matter how much computers learn how to do, it's how we come together as humans that matters.
That's all for this episode of AI and You. Please leave a rating and comment and share with your friends. Get the book Artificial Intelligence and You and see more videos and articles at AIandyou.net. That's AIANDYOU.net, where you can also send us your questions. Thank you for listening.